Hey everybody, we're going to look at this exam question today about the Osmosis required practical from Biology Paper 1. This is required for triple and combined students. So it's a long old question, so make sure you've got a bit of time to watch this or save it for later. A student investigated the effect of concentration of salt solution on the mass of uncooked potato pieces. So that's our independent variable. Mass of uncooked potato pieces is going to be our dependent variable, what we measure. So this is the graph of the results. So along here you've got the concentration of salt solution increasing up to 1 moles per decimetre cubed. And then you've got the mean change in the mass of the potato pieces. So here is a positive change, so they've increased in mass. Here's a negative change, so the potatoes have decreased in mass. So we have to plan a method that could be used to obtain the results for 6 marks. So we're going to go over the page, but this is a describe a method investigation type question. So we need to make sure we talk about 3 important things. That is the method you use, that is the repeats that you would put in place, variables you control. Those are the 3 most important things to consider when you have a question like this, plan a method. So let's do the method first. So the first thing that is that you're going to create your potato pieces. We would need to keep them all the same, but we'll talk about that later. Create the pieces of potato, place them in different concentrations of salt solution. I think to get these results, you would need at least 3, but I think 5 concentrations as stated on the graph would be better. You could do 3 if you kind of had values along this line, somewhere along there, and here. You could still plot a curved line, but the more concentrations you test, the better. So we're going to start with prepare potato pieces of the same size. Place in 5 different concentrations of salt solution. Now, those would be kind of like in test tubes. So this is your test tube. There's the salt solution in there, and then the piece of potato sits inside the salt solution so that it's covering it. And then you're going to leave those pieces of potato for a decent amount of time, overnight if possible, so 12 hours or more if possible. This just gives enough time for the water to either leave the cells or enter the cells. Leave the potato for at least 12 hours, and then you're going to remove the potato. It's important at this point, as you remove the potato after the time, that you place on some kind of paper towel or something absorbent, and that is just going to allow any residue of the liquid to be dried off, because we don't want to measure the liquid, we just want to measure the potato. Blot dry using a paper towel. What we didn't say, what we forgot to say, that we need to measure the mass before the 12 hours and then after. So we'll just put this in now, better late than never. So I've just said here, measure the mass of the potato piece before soaking and then after. We are going to make sure now that we repeat that for each concentration at least 3 times. And the reason you need to repeat it at each concentration at least 3 times is to be able to identify any anomalies and calculate a mean. So you'll now be able to work out the change in mass, the mean change in mass. So you'll have mean mass differences between before and after for each concentration. Calculate the change in mass for each concentration. That is what's been plotted here. So we must calculate that in order to be able to plot this graph. We've talked about the method, we've talked about the repeats. We now need to make sure we control all the variables to make the results valid. So we need to control the size of the potato pieces. We need to make sure we use the same type of potato, or even better, the same potato for everything. Different types of potato will have different types of cells with different water concentrations in potentially. So the type of potato. We need to make sure that we use the same level of blotting each time to get rid of all the excess water. Whether that is to coat the entire piece of potato in paper or just dab it a few times, you need to make sure it's the same for each single repeat that you do. we need to make sure that each of those concentrations of salt solution is kept at the same temperature. So that would be enough to get you full marks, because we've talked about the method, we talked about the repeats, and we've talked about the control variables. Next question, explain the results for the potato pieces in the 0.6 moles per decimetre cubed. So that's here. So you can see that the mean change in mass is negative by 0.2 grams. We have to explain why that is. So that's because the water has left the potato cells via osmosis, so it's lost mass of the water. The reason that water has left the potato cells is because the concentration of water is higher inside the potato cells compared to outside. So water leaves the cells by osmosis. Water has left the potato cells by osmosis. The potato pieces lost mass. This is because the concentration of water is higher inside the potato cells. Last question, explain why the result for the potato pieces at 1 moles per decimetre cubed was different from the result at 0.6. 1 moles per decimetre cubed, the potatoes lost more mass, close to 0.3 grams on average compared to 0.2 grams on average. This is because 1 moles is a higher concentration of salt solution than 0.6. Therefore, there is a steeper concentration gradient. There is a bigger difference in concentration of water or salt between inside and outside the potato cells. The pieces lost more mass at 1 mole per decimetre cubed because more water left the cells, and then we'll explain that more because there was a steeper concentration gradient. In other words, there's a greater difference in the concentration between the inside and the outside of the cells.